Mitochondria, often referred to as the powerhouse of the cell, are double membrane-bound organelles crucial for energy production. These organelles are found in nearly all eukaryotic cells and play a vital role in converting nutrients into adenosine triphosphate, ATP, the energy currency of the cell. A well-labeled diagram of a mitochondrion reveals its complex and efficient structure designed for optimal energy conversion. The outer membrane is smooth and encases the entire organelle, allowing the passage of small molecules and ions. It contains protein complexes known as porins that permit the entry of necessary molecules. Inside lies the intermembrane space, a region critical for various biochemical processes. The inner membrane is highly convoluted, forming cristae that increase the surface area for ATP production. This membrane is impermeable to most ions, contributing to the formation of an electrochemical gradient essential for ATP synthesis. Within the inner membrane is the mitochondrial matrix, a gel-like substance containing enzymes, mitochondrial DNA, and ribosomes. The matrix is the site of the Krebs cycle, or citric acid cycle, a series of chemical reactions that produce electron carriers used in the electron transport chain. The cristae host the electron transport chain and ATP synthase, the mechanisms responsible for oxidative phosphorylation and ATP generation. Moreover, mitochondria have their own DNA, supporting the endosymbiotic theory that they originated from ancient bacteria. This genetic material encodes essential proteins and allows mitochondria to replicate independently of the cell. Understanding the detailed structure and function of mitochondria highlights their indispensable role in cellular metabolism and energy production. Their intricate design and efficiency are a testament to the remarkable complexity of cellular machinery.